We would like then to move to our next series of speakers, and I would like to introduce Dr. Russell Poldrack, who is going to be representing the DPACC on these particular talks. Then we're going to have a talk by Dr. Robert Heinkel from the NIMH. And then we will have a talk from Dr. Dan Pandina, who is from Janssen R&D and one of our steering committee members and private partners. So I would like to turn it over to you, Russ.
All right, thanks very much. I want to talk about a sort of ecosystem of open tools for reproducible science that my group has been involved in developing and that is sort of feeding into the work that we're doing with DPACC. So, we all know that the ability to reproduce another researcher's findings is a defining feature of science, right? Going back to like the 1600s when Christian Huygens wanted to try to replicate Boyle's apparatus for pumping air out to see if there's a void in a void. But we also all know that this doesn't always work. And increasingly over the last decade, we've seen that, across many different areas of science, really sort of most prominently in psychology first, but clearly in lots of other areas as well, science doesn't always live up to this, this promise of reproducibility. So we might ask about, what in, neuroimaging is a particular field I work in. What do we know about reproducibility of neuroimaging? And we haven't had the kind of replication studies that we've seen in psychology, mostly because neuroimaging is really expensive. We can't just go out and replicate 100 studies. But there's evidence that certainly that reproducibility might not be great. And I think most strikingly is this paper that came out a few years ago from Mueller et al. They basically did a meta-analysis of previous meta-analyses that had looked at case control differences in depression, basically, comparing depressed and non-depressed individuals on a number of different functional MRI measures. Each of the previous meta-analyses had suggested that there were consistent case control differences. But this is, Mueller et al. Are sort of the best group in the world at imaging meta-analysis. They fixed some problems with the previous meta-analyses, and they basically saw that across those 99 studies, each of the 99 studies had reported a significant case control difference. There's no consistent convergence across those studies. So they all basically come out to nil when you put them together. That's worrisome, right? And there's a lot of reasons for that. I'm going to focus on one particular reason, which has to do with analytic variability. We know that in imaging, there's a large degree of flexibility in how we analyze the data. And we wanted, in a recent study, we wanted to know how much this matters. And so we did a study. This was really led by Tom Schoenberg's group at Tel Aviv with a bunch of other people who were involved in designing it, which basically looked at what happens when you give a bunch of people the same data set and ask them to analyze it, the same fMRI data set. So we collected, I'm not going to go into the details of the data set. We collected a data set of people performing a decision-making task, 108 subjects collected at Tel Aviv University. And then we gave out the data. We recruited teams basically to analyze the data of up to 3 people. They had to sign a non-disclosure agreement agreeing not to tell anybody else what they found until the embargo was lifted. And we asked them, we basically gave them about 3 months, and we asked them to give us yes, no answers on 9 hypothesis tests, which are of the form, is there significant activity in some brain region for some sort of feature of the experimental design? So for example, does activity in the prefrontal cortex, particular part of it, go up as the amount the person could win on the gamble goes up? And we told them to analyze the data as if it was a study in their own lab. 82 teams took the data, 70 teams gave us results back, and they gave us back their yes, no decisions along with a bunch of other data that we were able to analyze to look at sort of, what drove their decisions. And they also gave us a detailed written description of their analysis workflow using this best practices guide that we had been involved in developing a while ago. None of those 70 teams, no 2 of them used an identical workflow. So there were 70 different analysis workflows for 70 different teams. And even with those detailed written descriptions, it was often hard to tell what they had done. We had to actually ask for like code in some cases. Here's the main finding. This is showing the proportion of teams that either said yes or no to each of those 9 hypotheses, sorted by that proportion. If all the teams agreed, all these numbers would either be 0 or 1. That is, all 70 teams would say, yes, there's activity, or all 70 teams would say, no, there's not activity. And we see that for 3 of them, the first 3, roughly everybody agrees there's nothing going on. For one of them, most teams agree, yeah, there's probably activation, about 85% of teams. But then we have these 5 hypotheses in the middle where 20 to 40% of teams said yes. That's a striking amount of lack of reproducibility. Another way to look at it is across all the 70 teams, there were 33 different patterns of yes/no outcomes. And for any particular hypothesis, you can find 4 workflows that can give you a positive result, or many more than that can give you a negative result. We also looked at the whole brain data that they gave us and saw that if you look at each voxel and ask how many teams said yes, said this voxel was active, the highest overlap was 76%. So the voxel that was most activated only had 76% of teams saying it was activated. Nonetheless, we're able to meta-analyze across all the different groups. We actually find consistent activity. And I'll come back to this later because we think this is a really important point that, ultimately, we probably want to move away from using a single analysis workflow for any analysis to looking for convergence across many different analysis workflows, what people are coming to call multiverse analysis. So analytic variability is clearly a contributor to our reproducibility problem. We don't think it's the only one, but it's an important one. So how do we fix this? Well, we're sort of inspired by this idea that sunshine is the best disinfectant. And we think that openness is a critical element for reproducible science. And sort of inspired by this quote from a paper long ago by my colleague, David Donahoe. An article about a computational result is advertising, not scholarship. The actual scholarship is the full software environment code and data that produce the result. So we've been, along with many other colleagues who I'll recognize at the end, we've been involved in developing a sort of an ecosystem that's centered on open and reproducible pre-processing and ultimately statistical analysis as well of neuroimaging data. Again, starting with a focus on fMRI, but ultimately, generalizing out to other types as well. So I'm going to talk about a number of the different portions of this framework. We call it NIPREPs. The first, I'm going to talk in a bit about fMRI prep, which was the first of these tools, and we've subsequently kind of generalized out to other sorts of data as well. I first want to say a bit about what I mean by open. In the open source software world, people talk about 2 senses of open, right? Open, 2 senses of free. And so we want to make everything freely available, right? And by that, I mean both free as in free beer, like we're just giving it to you for free. You don't have to pay anything for it. And that also means you don't have to buy any other software. You don't have to go buy a MATLAB license or an SPSS license. It's all based on free software. But probably more importantly, free as in free speech, right? We give you the license to do whatever you want with the stuff, modify it in whatever way you want, and redistribute it however you'd like. So free in both of those senses. Open also means open to everybody. And so we really focus on trying to build, we don't want this to just be a project that our lab is doing. We want it to be a community. And so we really try to focus on building a welcoming and inclusive community. We do that through making it clear how people can contribute and by having explicit codes of conduct so that people are treated well when they're part of the community. This is an example of the contributing guideline from the NIPREPs project, where we say, there may be a little voice inside your head that's telling you you're not ready to be a contributor. We assure you the little voice in your head is wrong. So we really try to encourage people to contribute in whatever way they can. That might be writing code, might be helping in other ways. So let me start with sort of the foundation for this whole ecosystem, which is this thing called the brain imaging data structure, or BIDS. And I think Marty also mentioned this in her talk. So this is something we started developing back in 2016 that's become a community-based open standard for neuroimaging data. And that means it both describes how you name and organize files and folders, and how you organize metadata, which are the additional data beyond, say, the images that you need in order to be able to interpret what's going on. This started as an MRI-centric standard, but it has a mechanism for extension by people in the community. And that's actually been taken and run with. So we now have extensions for various types of kind of neuroelectromagnetic measurements like EEG and MEG, For PET, there's a microscopy extension. There's a lot of different extensions, and it's a really growing community. And you can sort of see that if you go to the GitHub page where this stuff happens. We have so far 71 people have contributed to the specification. That's a lot for an open source project. We have an elected international steering group that I'm a member of. I'll be rotating off this year. That has people from many different communities, different imaging types. We have 6 different maintainers who basically either give their time or are paid to basically keep the standard going, help ensure that it works for everybody. And we've had a bunch of meetings from people. It's taken a ton of work by the community to really develop this, but I think we've seen just how impactful it's been in sort of enabling people to share data really effectively. Now, whenever, putting a data set into the bid standard, it takes work. When it comes off the scanner, you have to sort of move it to a different format. And early on, we wanted to know, how can we sort of incentivize people to do that work? And so one idea that Chris Gorgolewski, who was sort of the inventor of bids, came up with was the idea that we should give people software tools such that if their data is in the bid standard, they can really easily apply the software. So for example, many of you are familiar with FreeSurfer, right, which is a package for computational analysis of structural MRI data. And if you just want to run FreeSurfer on a data set, you have to sort of reformat the data, put it in the format that FreeSurfer wants. But we worked with them to create a bids app. So now if you have a data set that's in bids, you just point that bids app at it, and it'll automatically run FreeSurfer on it without you having to change your data at all. And the other thing that these things do is that we use a technology that I'm not going to go into detail about, called containers, that basically encapsulates not just the software, but all the dependencies, giving you much better reproducibility, because you can know exactly what went into running that particular analysis. I'm going to talk about a couple of bids apps that our lab was involved in generating that are both going to be used by DPACC. The first is something called MRI-QC, which it basically does quality control on both structural and functional MRI data. The field struggled for a long time to have kind of really usable quality control measures and metrics for MRI data. And so Oscar Esteban built this tool a number of years ago that basically takes a bids data set and provides you with a really usable report on the quality of those data, starting at the group level, showing you, for example, distributions of a number of different image quality metrics. And then, say you want to dig into this particular subject's data, you can see the individual anatomical data. So it provides a really easily usable quality control report. Another thing that we try to focus on is, providing good documentation. And so Oscar and a number of other people from the community have worked on this, what they call the QC book, which is basically an open source guide for doing imaging quality control using MRI QC and other tools for both actually human and preclinical MRI. And so I think that that sort of documentation is really important. I'd recommend having a look at that. Another thing that we generally try to do is take advantage of people using our tools to get a little bit of information back. So when someone runs MRI QC on a data set, they're able to opt out of this, but if they don't opt out, a little bit of information, in particular the image quality metrics, along with a little bit of metadata, are sent to a database. That database is actually hosted at NIMH by Adam Thomas's group. And to date, this shows the growth in that database. We have quality control metrics from on the order of 375,000 unique fMRI scans and about 280,000 unique T1 weighted scans. And those data are all publicly available. You can download them from an API that's at that URL. These are just a couple of the kinds of analyses that one can do with those data, right? So let's say you want to know, how does temporal signal to noise ratio change with multiband acceleration factor for fMRI data? This is over those hundreds of thousands of data showing what that looks like. We also have, there's data in there from many different MRI scanners, right? We know that that's part of the metadata that gets stored. So let's say you want to know, what's the contrast, how does the contrast to noise ratio, which is a quality measure for T1 weighted images, how does that differ across different scanners, right? And you can see in this case that like the Philips and the GE seem to do a little bit better than some of the Siemens scanners for reasons that I don't quite understand. So that was one of the first tools that we built. I think the most impactful one is a tool called fMRI Prep. So you may or may not know that fMRI data require a substantial amount of pre-processing before one can ever do any statistical analyses. And that's both pre-processing on the structural data and the functional data, and then aligning them with one another, various sorts of artifact removal steps, and so on. And in most labs, this is basically done by deciding which package you want to use, FSL, SPM, AFNI, downloading that package, and then running that package's entire workflow on one's data. The goal behind fMRI Prep is, one, to basically say, well, we're not sure that any of those individual packages has all of the best tools for each step, right? So taking advantage of the NIPIPE workflow system, we're able to put together different, we might want to use the brain extraction from FSL and the alignment between structural and functional from FreeSurfer and something else from AFNI. So we're able to do that. The other thing that we do is we try to basically take advantage of the data set and its features to do the best possible job we can. So for example, if you have field maps, we will process those field maps and try to use them for distortion correction. If you don't, we actually offer a field map less distortion correction. We process the data. It's meant to be a relatively agnostic tool such that it'll give you whatever you need to go do your analyses without really any regard to what those analyses might be. So we do both surface analysis and volumetric analysis. And it was, this tool was, we say that it's robust because we benchmarked it on a large number of data sets to basically make sure that, from OpenNeuro, to make sure that it would run on all of them. And we've seen that it runs successfully. A danger with a toolkit like this is that it can be a black box, right? A person who knows nothing about fMRI preprocessing can apply this to their data and get results from it. So we like to say that we encourage a glass box philosophy with fMRI Prep, right? So giving somebody that black box, powerful black box, can be like giving a kid a Ferrari, right? That, bad things are likely to happen. So we do a few things to try to prevent that. We write educational documentation. We give people reports that help them verify and visualize the assumptions. And we also help them to sort of disseminate their results effectively. So here's an example. Hopefully you can see the animation here. This is one of the visualizations that helps people determine whether the alignment of structural to functional data has been effective. So we basically zoom back and forth between the T1 weighted and the bold image with the cortical surface overlay. So if something's gone wrong, a visualization like this makes it really easy to see that pretty quickly. And so we give a number of visualizations and reports that help people determine has anything gone wrong in the processing stream based on our experience of what particular steps tend to go wrong. One challenge that we know is that people often don't report their workflows properly. So another thing that we do is we provide kind of boilerplate text to basically tell people, here's how you can describe the workflow that was performed by fMRI Prep. For example, giving all the versions of software and so on. Marty mentioned earlier the FAIR principles. And we take a lot of inspiration from the FAIR principles and in all the work that we do. I'm not going to talk today about our data sharing work, but like the OpenNeuro project that we've developed is really inspired by those principles. One of the really important aspects of the FAIR principles is this, the need for community standards. And I mentioned BIDS before. Another project that we have been involved in there's a paper that's accepted pending some minor revisions at Nature Methods. It's a project called Template Flow, and it's really meant to address another problem that we've come across in the imaging literature. So one of the really important things we do in imaging is we generally align individuals' brains to some common template. Right. So there's various templates that have been developed by various groups. Many of them grew out of this sort of early work from the Montreal Neurological Institute. And so there's a number of templates that are often referred to as the MNI template. And if you use, any of three of the different software packages, you will be told that you're registering your data to the MNI template, but the actual template that you're registering your data to in each of those different packages is different. There turns out to be a number of different versions of the of this quote unquote MNI template. And also there's no sort of, clear way to sort of go find these things other than sort of downloading the packages. So what Oscar Esteban and Rasko Siric in my group developed was this package that basically provides sort of a database and a programmatic way to access templates and also to map between templates. If you want to move from one template to another, it makes it very easy to do that. Also provides a number of additional things like atlases that are associated with these templates. This is a demonstration of all the different templates that are in the database, mostly human, mostly volumetric, but a couple of rodent ones and a couple of surface ones. So this tells you sort of all the things that are contained in that database. Some of it is various images that have the template, various masks, various atlases, and then a bunch of metadata. One important part of the metadata, well, there's a couple. One is the changelog that basically says, how have things changed over time? Another is a license that explicitly says, what can you do with this particular atlas, right? We think it's really important that anything that you share, and this is part of the fair sharing idea, anything you share needs to have an explicit license associated with it that tells people what they can or can't do. Now, one of the cool things that the fact that this is sort of programmatic, right? There's Python tools that allow you to sort of pull these things from the database. We can now, if you're using fMRI prep, you can put your data into any of those output spaces by basically just saying, I want these output spaces. And Template Flow will pull them down. If you want it in 3 different versions of MNI, it'll let you do that. And so this is a tool that I think is going to become really important. Any software developer can use it to pull in and have really good version control over the templates that they use. Okay, I want to finish up talking about sort of one other effort that we've been involved in, which has to do with another aspect of what open means to us, which is that we should be collaborating rather than competing with one another. And we're not the first people to say this. Stephen Johnson has a nice quote from his book on where good ideas come from. Openness and connectivity may, in the end, be more valuable to innovation than purely competitive mechanisms. We're often better served by connecting ideas than we are by protecting them. And so we noticed a few years ago, as did a number of others, that a bunch of different labs were basically duplicating effort in order to build their own tools, right? We had fMRI prep, other people were developing versions of HCP pipelines, other people had other pre-processing tools. And so we started to ask ourselves, how can we go about reducing this redundant effort? And ended up developing a consortium that we call InMind, which stands for Never Mind, This Method Is Not Duplicated. It's the leaders of this group are Ted Satterthwaite, Damien Fair, Satra Ghosh, and Mike Millam. And Greg Keir from the Child Mind Institute has really been kind of driving the project. And really what it came down to is a bunch of us coming together to ask, how can we work together so that we can more easily share code with each other and trust that, that it all meets the same standards. So the guiding principles are really that we need to align ourselves on a set of standards, including coding standards, like we're all working in Python, right? So we have that kind of basis for the most part. What shared coding standards are we going to have so that if my lab gives code to somebody else or they give code to us, we can trust that we're equally happy with the quality of that code. And testing standards to make sure that it's all well tested. data performance standards and make sure that, we have benchmarks that can tell us where, whether things work or not. Terminology standards. So when we use a particular term, we're talking about the same thing. And finally, documentation standards. So we agree on how we need to document things. We're also developing some accessible sort of semi-automated tools to help people tell whether what they've done actually meets the standards. And ultimately, we want to get more groups than just this small number of groups to start using these standards, and ultimately, have a shared code base that we can all work with. So we don't have, 8 different groups developing, a DICOM to Nifty converter or something like that. So this is sort of, what we hope to have in the end is sort of a dashboard where, some tool like DPACC, will have been assessed across, the various different standards and ultimately get, does it get a bronze or a silver or a gold shield for each of those. So, so I just want to end by, saying that this isn't just something that my lab has been doing, that it really does take a village. And we've been really lucky to have a large number of people who have been, really invested in building this ecosystem and helping it grow. A number of them are here. There's certainly more than this, but these are many of the main players, some of them from my group, many of them from other groups. And so I want to end there. And I don't know if we actually have time for questions, but thank you very much.
Thank you very much, Russ. That was a fabulous talk. Very impressive. So we have time for a few questions. I guess a question that I have for you, Russ, that maybe you can't answer, but I mean, this looks like such a wonderful system that is in place. I mean, what do we have that's comparable for other types of data? I mean, imaging has been, really a core technology for a long time, but I mean, we have other technologies coming down the road that could really benefit from the learnings and experience from you and your groups.
Yeah, I think that, we certainly are ahead of the curve. And, so for example, people in the behavioral realm are developing standards kind of inspired by BIDS. There's something called PsychDS that, that's being developed by Melissa Klein over at MIT. BIDS is also being extended to other types of data. So for example, there's an eye movement standard, an eye movement extension for BIDS right now. Yeah, and certainly there's other people in other domains are building standards. So for example, there are other groups, outside of BIDS developing standards, say for wearable, data from wearables and things like that. I think that certainly what we've done has inspired others, but we definitely are ahead of the curve in imaging.
Thank you. We have a question from Joseph Cambites. Would you like to say your question?
Yes, yeah, thanks, Mr. Poldrack, for this very inspiring talk. My name is Joseph Cambites. I come from Germany. I'm part of the Prescient network. You mentioned the multiverse analysis, if I understand correctly, but that this term refers to an analysis where you would sort of do all the possible workflows and then see how the results change depending on the workflow. And you mentioned that this is an area that you think the field would move more and more towards. Right. I'm not sure how this would be like implemented in the procedure of publishing results, and also how this would work out in the context of very complex analysis like machine learning or the computational modeling approaches that we heard about. Could you elaborate a little bit on your ideas here? That would be very interesting.
Yeah. So sorry, I had meant to put a slide in about some of the work that we're doing on developing tools for multiverse analysis in the context of imaging data, and I ended up pulling that out. So I think those are both really good questions. In terms of how it gets reported, the way to think about it is it's sort of like doing a meta-analysis, right? In fact, the tools that we used in that one slide that I showed for the NARP study, those are really kind of meta-analytic tools, right? That say basically what's consistent across all these different workflows. And in imaging, we're not going to be able to do thousands of workflows as people might do in some of the behavioral work. But we could certainly do, say, 50 or 100 kind of reasonable workflows and ask, what results are consistent, or, it might also be interesting to know, like, what results vary in a direct way with particular aspects of the analysis, because that might tell you something about, what's going on with those analyses. There's no reason that you couldn't also do this with, with things like machine learning analyses. And I think it might even be more important there. So, so there's no, for example, let's say that you want to do, classification, right? There may be no, like, intrinsic reason to use, say, like a, lasso versus, L2 regularized logistic regression, or, any of a number of support vector machine, whatever, right? So you kind of like to know, like, what's consistent across. If there's no reason to choose one of the analyses, it'd be nice to know what's consistent across them.
Well, thank you very much for that question and for that great response. Really appreciate it.